day. So today's topic of interest was role of flow cytometry in medical biotechnology as an overview. My topic of the talk is about this. So flow cytometry is a technique which is an advanced technique. So in this presentation, I'll be discussing about the advances, the technique, the clinical impact, and its importance for disease biology. So as I start now, what is as I have already said that what is the flow cytometry? So flow cytometry is a biotechnological technique or a biomedical technique or just a biological technique which is used to detect or measure the physical and chemical characteristics of a population of cells or particles. It can be cells in case of living organism or it can be particles in case of viral particles or the viruses which are not comprised of a cellular shape and structure. So why it is said to be a flow cytometer? Because if we break down into its components, it becomes as flow cytometry. There are three parts of it. First is flow, means the cells in motions. Now, when we go for flow cytometry, what initially strikes in our mind is that the flow of cells, the cells which are flowing, and the flowing has a motion. It is hello. It is in motion. Flow relates to cell and metry means measurement. So it is not just the qualitative analysis, but it is the qualitative analysis of different types of cells or the group of population or the population of cells. It is a enumeration of the number of cells or the types of cells. So it is a multi-parametric analysis of the cellular components. Now, when we come down to the flow cytometry, means it is the measuring the properties of cells while in a fluid stream. So it is a dynamic method of measuring the cell population or seeing the physical chemical properties or the characteristics of a cell which are in motion in a fluidic medium or in a continuous flow through a channel. Now, this is just a general view of the advanced setup of a flow cytometer. It can be now it is becoming a benchtop flow cytometer. It can be a full length flow cytometer. There are different companies which are making flow cytometers of different kind. So there can be a benchtop flow cytometer as well as in earlier stages of early development before two decades. This flow cytometer was capturing a whole room as well for a setup. Now, coming on to the basic principle. Now, what is the basic principle of flow cytometry? It is the basic principle of flow cytometry is the passage of cell in a single file in front of a laser so that they can be detected, counted, and sorted. Now, the three properties which I have written, that is, one is that cells can be detected based on their properties, means which type of cell is present, that is a detection. If there is a heterogeneous population, if there are quite a big number of cells, there is a cellular population where there are around 20,000, 30,000 cells is present there, so that will be detected on the basis of their nature, on the basis of their anatomy, on the basis of their cellular structure. Then it will be counted, as I said, what is the number of cells, and then it will be sorted. So that can be sorted as well. Now the sorting and counting and analysis, there are two types of parts that can be divided by a flow cytometer. I will come to this later on also. One is fax. You might have heard, easily heard, that is fax flow cytometry. Many people call it as fax. That is not fax, it is FACX, fluorescence activated cell sorter. So that is a sorter that is basically to differentiate cells based on their detection. Like it very common principle and very common application of it is in the HIV testing, where we have a population of CD4 cells and CD8 cells. So there basically the flow cytometer is not in the analytical part. It's just as a cell sorter part. So basically you have to put the sample and it will get differentiated into CD4 and CD8. So that is flow, that is cell sorter. Then the later advanced analysis comes to cell analyzers, where they will analyze the property, they will analyze the surface marker, the integral properties. Now, coming on to the cell components. What are the cell components? The cell components are fluorescently labeled they and then excited by a laser to emit light of varying wavelength. Like it is already saying that it is a flow cytometer or a fluorescently labeled. What is fluorescence? Fluorescence means to emit light or to emit any kind of light which can be which can be irradiated or can be collected via acceptors or diode. It can be collected, and it forms a graph or a histogram is maintained which shows different wavelengths of light, from visible light to UV light to different kind of light have have is being placed on the receiver, and it forms an image in the form of a graph or in the form of a chart where we can assess how the complexity of the cell by the form of these lasers emitting wavelengths of different frequency. Now how it works, the cells are suspended, as it is saying that flow comes from the word fluid. Now the cells are suspended in a fluid and flown through a laser beam. Now the laser being fluorescently labeled antibodies. Now the antibodies which are being prepared, which are being procured from outside, they are being like there is a basic principle of ELISA. That ELISA is a colorimetric test. This is a fluorescent based test. So the basic difference there is a is this like we are saying that flow cytometry, these are for cellular population. There is also histochemistry, that is immunohistochemistry. So these techniques are basically being defined on the basis of their cellular structure, on the basis of what kind of analysis we are performing. So in ELISA we are doing antigen antibody reaction, we are doing it in a just for a this this purpose, that we check for the color thing, and here we are checking it on the basis of light intensity and specific cellular populations based on which kind of cells are there, which kind of markers are developing, and what the difference from histochemistry and cytochemistry, because this is about the cell. So this is about cytoanalysis or the cellular analysis. So in this we will study not about the whole histology or about the whole tissue sampling, but about particular cellular properties. So the fluorescent label antibodies bind to specific markers on the cell. Now, what are the markers? There are biomarkers. There are many kinds of these antigens or the kind of surface receptors that express on the surface of the cell. Now these receptors bind to different kind of antibodies, different kind of drugs, different kinds of pathogens. They bind, and this binding makes the cell activated or may elicit any kinds of response from the cell. So this when this kind of interaction happens and there is a fluorescently labeled antibody, this fluorescent or a fluorophore which is being attached to the antibody gets fluoresced, and it is being recorded on the system. So now, what is the basic advantage of having this flow cytometry is that it allows rapid detect analysis of thousands of cells per second. As I initially told, that it is based on the detection, count, and sorting principle. So the count of the cells, if you can count 20,000, 30,000 cells, there is a group of population of cells. It can be counted rapidly within a short span of time, and it can be separated based on a different analysis. Now what is the, as we our talk says, the role of this technique or this biotechnique in medical biotechnology. So what is the role of this flow cytometry in medical biotechnology? It is essential for cellular analysis, particularly in diagnostics and treatment monitoring. So as I said, that flow cytometry not only aids in diagnosis, it will also assess in treatment monitoring, like after we give a drug or we give some immunotherapy or we give some any kind of cross matching is done for blood transfusion, different kind of analysis or medical procedures, biomedical procedures are being done. So this flow cytometric analysis can aid us, can help us in seeing what after effects of after treatment or the post treatment, post surgery, post transplant, what are the changes that is being occurring in the system or in the analysis, and how the our body or our cells is responding to those different kinds of treatments. Now, what happens in a flow cytometer? As I was telling you, you might have got an image at the behind side of your brain. Now, that can be seen by this figure also. There are the three components. The three components which makes this a flow cytometer is flow. As I was telling you, it's about the fluidics. Now there are three parts: fluidics, optics, and third is the electronics. Now the fluidics part says that if the cells are in a suspension and flown in a single line through a illuminated volume where they scatter light and emit fluorescence. Now the cells under a sheath fluid are passed linearly across this mm, across an illuminated channel, and a laser, sorry, I was forgetting that word, that it is flowing by a laser, and that laser emits different kind of fluorescence or different kind of intensity colors, which light which scatters and being collected by these receptors or those diodes, which comes under the optics session. Now the optics part is about the collection of these light or these scattering in different intensity lights. These are being converted by the diodes or by the photomultipliers which multiply them, and these are connected onto the system via the electronics to digital values and are stored on a computer in the form of a representation or analysis is being done by different kinds of softwares. Now, the basic components of a flow cytometer, as that was the hardware part with that how the structure of flow cytometer is being done. Now, the part how the analysis is being done is, I was telling that the cells are suspended in a sheath fluid, which ensures that there is a fluid or there is a medium in which the cells, like the cells of the bone marrow, the cells of the this brain aspirate, there are different kinds of, or the blood, these are suspended in a sheath fluid, which makes the flow of the cells in a way ensures single file cell passes through the laser. Then comes the lasers. The lasers or the light beam or the source which is used to excite the fluorophores or the fluorescent fluorophores which are being attached to the antibodies bound to these fluorophores which are bound to the cellular markers, these get bound, and when the lasers excite these fluorophores, they emit light. Then there are detectors. There are different kind of detectors. There are basically, which I have made simplified into three types. One is the forward scatter, the second is the side scatter, and the third is the fluorescent detectors. Now what is forward scatter? As as the cell flows, the size of the cell gets measured by this forward scatter. The light beam passes, and the size of the cell is being measured by this forward scatter. Then the light which gets through the cell goes to side scatter. Side scatter measures the cellular granularity or the complexity, means how many nucleus are present. This differentiates different kind of monocytes, lymphocytes, the blood, different kind of blood and cellular components are being differentiated by on the basis of side scatter. And then there are fluorescent detectors. These measure the emitted light from the fluorochrome labeled antibodies. Then finally the system which is recording these all fluorescent light from the fluorescent detectors, and they are capturing it. There are softwares which analyzes the data for a specific characteristics, like I'm telling you that it is about size, it is the granularity and the fluorescence. Then uh, then the principle of flow cytometry means this granularity, this size, this structure, these fluorescence, what all these interpret, what are these presenting. So these present, as initially I was telling you that there are three major properties of this flow cytometer. One is detection, the other is cell sorting, and the third one is cell counting. Now the cell sorting, which if we go to the cell sorting part, so cell pass through a laser beam and light is scattered and detected. The forward scattered, initially we told that it is about the cell size. The side scatter is about the internal complexity, which kind of organelles are present inside the cell. What are the granularity of the cell? What is the DNA present in the cell? The cell is in which phase of cell cycle? Is it a apoptotic cell? Is it a diet cell? Is it a functional cell? Which kind of cell is present? That is. Then fluorescent. Then with the fluorescence is about the fluorescent antibodies which bind to specific cell proteins. As the cell surface markers are there, like the CD markers, which are well known. Like we talk in HIV, we talk about CD4, CD8. Then we have cancer markers, NK cell markers, dendritic cell marker, different kind of immune cells, B cell, T cell. They have different kind of markers. They are being irradiated, they bind to the fluorophores and they fluoresce and get detected. Now, what is the best part of or the principle of this flow cytometry is the multi-parametric analysis. When we do different kind of analysis, we get a single parameter. We analyze the cell on a single parameter, either it's immunohistochemistry or it is different kind of other techniques, histological techniques. We look under the microscope. We see the single dimension or a single parametric analysis. While in the flow cytometric, we get a 3D view or the 3D multi-parametric analysis. That is why we can get the size of the cell, the granularity of the cell, the fluorescence intensity, and these are being measured simultaneously. So we get to have less time, accurate prediction, and it can give you more and more detailed analysis of the system. Then comes the fluorophores. We have been constantly listening in the past few slides about the fluorescent antibodies. It gets fluorescent activated. So what are these fluorophores and their role? Now the fluorescent molecule that is being attached to the antibodies that bind is specific to the antigens on the cell surface or intracellularly inside the cell. They go and bind to some DNA molecule, or they form a probe. They become attached to any kind of tumor marker, or any kind of immune response. They get bind. So these fluorescent molecules are known as fluorophores. Now the common fluorophores are, which we are listen which are used as a single dye, single cellular based dye, are quite common, and it is largely being used in flow cytometric analysis is FITC, that is the fluorescein isothiocyanate. Then there is phycoerythrin, which is known as PE. Then there is L-phycocyanin, that is APC, and the pyridine chlorophyll protein, which is PERCP. These are quite common. Most of you have either ever heard about flow cytometric data. You might have seen a graph where there is x-axis and y-axis, and you might have seen that for H or S is written, that it is side scatter or forward scatter. F or S is written, and you might have seen this dye is mentioned. If you read any kind of papers, standard papers, you will find FITC, APC, APC, PE, PERCP, these kind of dyes are being mentioned. Now, what is the excitation and emission of these fluorophores? Now, these fluorophores absorb light and the laser from the laser and emit light of a specific wavelength. You can see a diagram also which is present beneath this slide. Now, different wavelength, when we have a white light, it produces 7 colors or different kind of colors based on the different wavelengths. So as the excitation of this fluorophore gets, and it emits a light in a range of 408 nanometer, or it is a 500 nanometer, or it is a 600 nanometer, different as different intensity of light is being emitted, and it is being captured and reflected on this screen. Based on this based on this graphical analysis, we get a result. Now, what is the purpose? The purpose of this analysis is to enable the identification and quantification of the specific cellular populations. Then the clinical application, what the most important part, what we now previously we have been discussing about the flow cytometer, or the component of the flow cytometer, the basic structure of the flow cytometer, how it can detect data, or how it can emit light. It is flowing linearly. These all were the previous analysis. Now, what are the clinical applications of flow cytometry? The clinical applications or the medical applications which is being directly translated into patient diagnosis and patient treatment. That is why medical biotechnology becomes more and more of importance, because these kind of biotechnological techniques, or the techniques which are being used in biology, are directly becomes translational research or translational medicine, which are directly applicable towards clinical medicine. Now, as we were talking about flow, so there will be analysis of fluids basically. So we have to make the sample in the part of fluid. So fluid detection will be in the part of hematology and cancer diagnosis. So hematology is about blood, which as it is talking about hemoglobin. So it is about blood. So blood and cancer diagnostic is about blood cancer. Then blood cancer is about basically is about two parts, that is leukemia or lymphoma. Lymphoma is about the lymph nodes, or the lymph is also, apart from blood, the lymph is also flowing as a fluid in our body. The most important principle what we commonly listen, these are the terms I'm talking about a basic talk, which is not as much detailed. If you want to go for initial analysis of any leukemia phenotyping or any kind of analysis, then you have to get trained for 7 to 8 days about a single topic, and that will be more detail oriented, that will be more about the software, about the analysis part. But here, as my topic is about, I cannot explain each and every specific disease and the diagnosis of it particularly. So that will take a lot of time. That will take, 2 3 days or this kind of thing for a session to go on. But as of overview, I can give you what is the application of it. So the immunophenotyping, because we can see it, we can see the colors, we can see the different kind of this graphical representation, the histogram, the pie charts that shows us the immunophenotyping, which is the major property of flow cytometry. So immunophenotyping gives us how to classify leukemia and lymphomas based on detecting specific markers. As previously I told, that surface marker, if CD19 is present on B cell or CD3 is present on T cell. So this can be differentiated based on the immunophenotyping, because these are immune cells for like B cells and the T cells. So these immunophenotyping will differentiate whether it is CD3 for T cell or whether it is CD19 for B cell, and that will initially differentiate whether it is a leukemia or a lymphoma. So then coming out from a mixed population, and then if we talk about minimal residual disease, as I was talking about, that if it is not only in diagnostic, it will also test the response or the quality of life, or what the drug or the post surgery, if you have been given a chemotherapy or you have been given a radiotherapy, any kind of therapy, you have been immunotherapy, any kind of therapy, if it is being given to you, it will also tell you the residual cells or the cells which are being left post treatment. So this can also, this is known as minimal residual disease. So that can also be detected by flow cytometry. Then comes about HIV and AIDS monitoring, which is immune deficiency disease. And it previously, as initially I told you, that flow cytometry is divided into two parts based on cell sorting and cell analysis. So in case of HIV/AIDS, this flow cytometry can be said as PACS. This is, I've written in the brackets as FACS, fluorescent activated cell sorting, FACS. So FACS differentiates the cell based on CD4 plus T cell count. This will differentiate that if the cell is CD4 plus positive, it can be treated as HIV/AIDS. So CD4 plus T cell count. Now, stem cell transplantation. What is about stem cell? Nowadays, it's all about stem cell and transplantation. We are using lots of studies for cellular transplantation, tissue transplant, even organ transplantation, blood transplantation. So this flow cytometric analysis is very much important in transplantation immunology and immunogenetics. This is a field which is there, which uses flow cytometry as a major instrument for their analysis. So what is about now, the transplant is about whether it can be a cellular transplant, it can be a tissue transplant, it can be an organ transplant, or it can be a blood transplant. So for that we need matching, we need donor and host matching, whether their cell or the components, their antigens are matching, antigen antibody, is there any kind of reaction, or is there any kind of means hypersensitivity. So that can be assessed by this, that whether these surface molecules are being assessed by flow cytometry. Now counting the CD34 plus stem cells in transplant patient to evaluate en- engraftment success. Now whether the graft has been done, if the CD34 plus is evaluated, and we can see that whether the success of the graft or the success of the transplant has been done or not. Now, flow cytometry in immunology, as previously also in case of HIV, there are different other diseases also which are present, and the immune response of the body, as soon as we give any kind of antibody or any kind of infection is there, the immune system of our body is the primary defense mechanism. Now, to analyze whether our immune system is hyperactive, it is performing well, or it is not performing well, we can assess by flow cytometry. Now, the primary immunodeficiencies, diagnosis of inherited or acquired immunodeficiency, like severe combined immunodeficiency, which is SID, and on the leftmost corner, the diagram is also presenting about SID. So, this diagnosis can also be done by flow cytometry. Then, allergic testing. Allergy is something which is present in every human being. Each and every human being is allergic to any of the pollen grain, any kind of environmental stressors, any kind of. So allergology is a specific branch which teaches about allergy or our natural reactions towards any kind of antigen or towards any kind of extracellular pathogen, any kind of it can be any kind. We can be sensitive towards abiotic factors, we can be sensitive towards biotic factors, some maybe to particulate matter, some towards light. So any kind of reaction where the immune system of our body gets activated is known as allergic response. And the basophil activation test. Now there are cells in our body which responds as a part of our immune system, like the basophil, the eosinophil, or the monocytes, the lymphocytes. These are so the basophil activation test is majorly used to detect the allergic reactions. Then the autoimmune disease previously, which one of the not the autoimmune disease. There are the different kind of autoimmune disease. So cytometry can identify abnormal immune responses, aiding in the diagnosis of diseases like there are systemic lupus erythematosus, rheumatoid arthritis. There are other autoimmune diseases as gout. There is skin diseases, autoimmune skin diseases like the this I mean autoimmune skin diseases are also there. Many kinds of autoimmune diseases are present in the body. Then comes the flow cytometry, where the most important aspect of flow cytometry, if we go on with numerous applications, we have two major applications of flow cytometry. One is in oncology, that is in cancer biology, cancer medicine, and the other is in transplant medicine, as I was telling you previously, that if we transplant any kind of tissue, organ, blood, any kind of transplant, or if there is a donation of any outer substance inside our body, that is comes under transplant medicine. So the if we talk from the left side, that flow cytometry in oncology, circulating tumor cells. Now, most recently, that is the liquid based cytology, the tissue cytology or the FNAC based cytology. These all have been excluded because these take much time and they are not as accurate as this kind of analysis. Now, FNAC is fine needle aspiration cytology. If we have a tumor, or if there is a swelling on your body, there are needles which take the aspirate from the this tumor analysis, and cytochemistry is done on the slide. So this is immunocytochemistry, that is ICC. And when we take the biopsy or a tissue from a tumor outside, and these both are invasive procedures where we invade, where so the non-invasive process, what is the non-invasive process? If we take about or we try to get a not we are saying that it is a non-invasive process, but least invasive, less invasive. We get a blood sample and we can see whether the cellular, which are these tumor cells, whether they are circulating inside our body, whether it is circulating in the lymph or whether it is circulating in the bloods. We can even not even the cells. We can have circulating tumor DNAs, circulating tumor RNAs. Small molecules can also be detected, or the particles can also be detected, which can give us a analysis, or we can get a previous information whether our body is prone towards tumor or cancer in later stages of the life. So that can be a good prediction for our susceptibility towards a particular disease. So detection and characterization of circulating tumor cells in patient with solid tumors like breast, prostate, we can get to know about what is the staging, what is the classification, where what is the prognosis of this cancer, whether this is going to relapse, whether it is going to reoccur. So these all we can get to know by this flow cytometric analysis of circulating tumor cells which are being circulated inside our body, and it has not means it we can say that it is in a pro tumor formation phase. It is in a phase where it will form a precancerous or a pro apoptotic phase or a phase. So then about the tumor heterogeneity, as initially also we were talking about that this flow cytometry can differentiate the population, sort the cells. So the sorting is done from the heterogeneous population where we have mixed number of cells. Now the flow cytometry helps us to understand whether the tumor heterogeneity by analyzing different cellular population within the cells. Now there are different markers. There is HER2 as a marker, human epidermal growth receptor factor. There are P53 markers, which are the guardian of the genome. So different kind of cellular markers are present, which get upregulated or downregulated by this tumor, and the different kind of cells which are which makes the tumor microenvironment or the tumor niche. This environment has different cellular populations. The migration of those cells towards the tumor, whether they are acting or not, whether they are making make the different kind of cellular pathways activating it, or the cellular communications, the cytokines, the interleukins, how they are releasing, how the cellular communications is being done. This tumor heterogeneity helps us to know whether the cells in the tumor population are actually cancerous or are in which stage of development. Then if we are just or talking about diagnosis, then comes the treatment part. There is a huge amount of means work is there about the cancer immunotherapy. Targeted therapy has also come to our place for cancer, but the immunotherapy or the classical immunotherapy is also there, which is about monitoring the immune population, because sometimes the tumor microenvironment or the tumor immunology is a study which is being conducted as of the most of the part. So first we try to see whether the immune response can be suppressed or can be controlled for later and later treatment by targeted therapy or not. So sometimes monitoring the immune cell population, example the T cells, the CD8, the CD8 T cells, cancer immunotherapy is being seen as in the development of cancer vaccines. So the vaccines which are being developed in case of infectious diseases, only infectious diseases, are also being developed in case of cancers also. So in case of cancer immunotherapy, we are trying to develop our global biotechnology industry is looking for developing this cancer vaccines also in the form of immunotherapies or the monoclonal antibodies, the MABs. So there are different kind of monoclonal antibodies which are being used in the treatment of immune regulated cancer or the checkpoint inhibitors. So these are basically the immunotherapies are the monoclonal antibodies which are used as inhibitors. One are the targeted drug therapy which destroys or kills the cell. One is the inhibitor, means if the surface of the cell is expressing some marker which needs some molecule which gets bind to it, and they produce signal for cell to become cancerous. This signal or the process of binding of effector molecule and the receptor molecule is being inhibited by these monoclonal antibodies. Some well-known monoclonal antibodies are trastuzumab, pembrolizumab. These kind of antibodies are there. Where there is MAB is written, it is an antibody. Then comes about flow cytometry in transplant medicine. When we talk about transplant medicine, one is the pre-transplant and the other is post-transplant. So before transplanting, we do a cross matching. Like it is a common process. We see when we need blood from a blood center or blood bank, we go there. The person appointed there does a cross matching. He uses our blood and says what we commonly know is that we say that we need a blood of this group particular. So that blood group A positive, B positive, AB positive, O positive is also having that Rh positive antibody or Rh negative antibody. So that needs to be cross matched with different blood which is being given. So we that blood which is being given from a different donor of if suppose if it is a positive and we need a positive, so that will be matched with the recipient blood sample. We take 1 or 2 ml of sample of the recipient which needs blood donation, blood transfer, that is being cross matched. So that determines the compatibility between the donor and recipient to prevent graft rejection. So that is being done before pre-transplant. That is being done for organ transplant also and tissue transplant also. Like there is live cell donor transplant in place of liver transplant. Then there is post transplant monitoring. What is post transplant monitoring is when we transplant or when we transfuse blood or any kind of thing that detects the early signs of graft rejection, whether our immune system has accepted our this transplant, or it is showing some immune response, because sometimes there is a delayed immune response or some kind of delayed hypersensitivity is there. It is not an acute response. It can be a chronic response or can be a delayed response. So the post transplant monitoring detects early signs of graft rejection by monitoring immune cell activity and identifying activated lymphocytes. Then the most important part, what are the advantages of flow cytometry at medical biotechnology? Why this flow cytometer become one of the ace technique or indispensable technique in the world of medical biotechnology? Why we are using it? It means, if I say as part, if you are talking about my part, I would say that real time PCR or RT-PCR, a flow cytometer, two instruments. These two instruments can make me do numerous kinds of tests. These two instruments only. You don't need to have n number of or 10 number of instruments. These two, if you give me a flow cytometer, if you give me a real time PCR, I can do most of your analysis, most of your tests can be done by these two instruments. Then if you want further, you can have some HPLC or a GCMS. These also can support. But for as far as this one, now the recent talk about medical biotechnology is about NGS, that's next generation sequencing, and the sequence analysis. That is also an integral part of our analysis. But flow cytometry is still is the classical way of analyzing in medical biotechnology. Now, what is the advantage? Is that high throughput. What is high throughput? That is, it can analyze thousands of cell per second. So that presents a robust data and a tremendous amount of this turnover time or this analysis, which makes us take clinical decisions, or the clinico-pathological decisions or the medical decision. The clinician is ready very fast. It is rapid detection. If he can take or she can take any immediate decision on any kind of treatment. Then multi-parametric, as I was telling you, that it is not just a single parameter, it has multi-parametric. It measures multiple characters of a cell simultaneously, not differentially but simultaneously. So that gives us a differential diagnosis. It aids us in having a differential diagnosis, means about the cellular size, what is the size, the granularity, surface marker. It the third thing, it is quantitative as well. So not about the quality or the size or the structure, about the quality of the cell. It also gives us the quantity, provides the precise data on cellular populations. Then about the so quantitative as well as qualitative. When we talk about PCR, there are two parts. We have PCR, which is the initial PCR, that is the reverse transcriptase PCR. It was having two parts, that was qualitative analysis. Then came the real time qPCR, that is the quantitative. So there are two types of it. But in flow cytometry, we have both advantages. We can have qualitative as well as quantitative analysis about the number of cellular population as well as the quality of cells. What is the quality of that cell, whether it is disease, whether it is healthy, any kind of information we can get. Then about the sensitivity, because in medical science or in medical biotechnology, two aspects is most important for diagnostic or treatment. One is the sensitivity and one is the specificity. So whether this analysis is specific, we have been talking about for the past few minutes, that it is very specific. It selects the specific population of the cells. And now comes about the sensitivity, about how much time it is sensitive, whether it is detecting at a rapid pace or it is taking at a low pace, or it is detecting it not as sensitively as others. So this detects the cellular population very sensitively, and making it usual for minimal residual disease monitoring. So it can detect even the minimum number of cells if it is present. It can sensitively detect. So that allows us about the prognosis of the disease, whether that disease will relapse, that will come again, or what is the lifespan or the quality of life of the patient, whether this patient will survive for 5 years, 10 years. That analysis is being done on the basis of the sensitivity of this test. Then on the right side, you can see there is a diagram which is showing different advantages of flow cytometry, or the different aspects of flow cytometry. It can be used in HIV therapeutics, it can be used in screening. Not only in this medical biotechnology, it can also be used for biotechnological research as well. It can be used based on the cellular analysis. Based on these analysis, we can screen bioactive agents, bioactive genes, which genes, as I said, which cells or which genes are activated, which genes are upregulated, which genes are down regulated. Biomarker discovery, small molecule means RNA, RNomics, this genomics, RNomics, proteomics. It can also detect the protein expression as well, drug action, drug safety assessment, drug efficacy, immunogenicity testing, pharmacokinetic analysis, antimicrobial drug discovery. So these are all most of the part. That is why I said that if one has to ask me my personal choice, I would say that if in a medical biotechnology lab or in a diagnostic lab, there should be one flow cytometer and one real-time PCR that can aid you a n number of hundreds or 200 number of tests. You don't need to have 10 or 20 kind of instrument. These two can make you do numerous kinds of analysis if used efficiently. Now, what are the limitations? Since we are talking about the most and more positive of this analysis, what are the limitations and challenges? So there are few limitations and challenges that are being presented by the flow cytometry. One is the cost. Since this instrument is not frequently available in different kind of labs, is the cost. This instrument is expensive, and the reagents or the dyes which are being used is also very costly. Sample preparation. Now the sample preparation is also a very tedious process. This requires a careful sample handling to avoid cell loss or cell degradation. Because if we did not get the sample properly, the analysis of our sample will also get hampered, and that can eventually cause a lot of harm to the patient and to our society as well. So the third point which comes is the expertise. That is why we need skilled personnel needed for operation and data analysis. So we need skilled person for sample collection, for the operation of this instrument, and finally for data analysis. So as the initiative of our country is also there, that Skill India program. So skill is the major requirement to handle these kind of instrument. Not only the skill in handling the instrument, but the soft skills or the analytical skills. Basically the analytical skills, how we are thinking of these results and how we are correlating with different kind of challenges that we are facing in any kind of disease diagnostics or treatment. Then comes the cell viability. What is about the whether the dead cells can interfere with the data accuracy, because the as we check for the apoptosis or we check for the live cell analysis, there is also a significant amount of debris or dead cells which are present in the sample. So that can most of the time either it is interfering with the results, either it is causing a synergistic effect, or it is causing an antagonistic effect. That also needs to be validated in our analysis. Now, the flow cytometry, which I was talking for the past few minutes, or about 45 minutes or 50 minutes, was that the classical flow cytometry. Now, the flow cytometry has become to much more advanced level. There are few more advanced levels that is being presented here. One is high dimensional cytometry. Techniques like spectral cytometry allows for the use of more fluorophores. What fluorophores? FITC, PER-CP, allopectin, AP, uh, these kind of these kind of dyes were present. They were single color dyes. They they use single parametric analysis. Now there are different kind of fluorophores like Alexa fluor, star bright, tandem dyes, fluorescent proteins, which makes simultaneous measurement of multiple cell markers. So these are multicolor flow cytometry. You might have heard the term multicolor flow cytometry. That Alexa flow, star bright is nanoparticle based dyes are being made. Then tandem dyes are two dyes working simultaneously. We use per-CP or FITC, two dyes are combined and they are working simultaneously to produce different color. Then there are fluorescent proteins. One very important fluorescent proteins which is quite common in biotechnology is GFP protein, green fluorescence protein. This green fluorescence, and there are other proteins also which is being developed, which are serving as a marker, which are used in flow cytometry for analysis. Then comes the mass cytometry, that is the CyTOF. CyTOF is the mass cytometry. This uses metal isotope instead of fluorophores to increase the number of parameters that can be measured without spectral overlap. Third is the single cell RNA sequencing combined with flow cytometry. As I was telling you that now, apart from PCR, the next technique becomes the flow cytometry. The third technique, which is burning, is about the sequencing. We talk about RNA-seq, we talk about DNA, this RNomics and transcriptomics or the protein analysis. Then this single cell analysis is also there. The single cell RNA sequencing combined with flow cytometry is also present. This allows both phenotypic and transcriptomic analysis of the individual cell. Now, what is when we talk about genomics, it's about the DNA, and when it is the DNA is transcribed to replicated to RNA and RNA is transcribed to protein. That analysis is also analysis, and that aids in this in the disease world, which we talk about. This epigenetic modifications or the epigenetic modulator. So this gives us analysis whether the phenotypic analysis or the transcriptomic analysis of the single cell RNA sequencing. Now the future directions in medical biotechnology. What will be the future of this medical biotechnology using this flow cytometry or the advances in future this flow cytometry? It is about personalized medicine or the individualized medicine. Now the this personalized medicine or the individualized medicine is of very much importance. We cannot have a single pill for a single person. Each and every person is having a different genomic composition or a body is differently built. So one drug can have a better effect on one person, and it may not be having the same therapeutic effect on different person. So this flow cytometric data can guide personalized treatment plans by identifying specific cellular population involved in disease. Immunotherapy monitoring, increased use in tracking patient response to immunotherapy and identifying biomarkers of treatment success is immunotherapy project. Then the most talk of or the most sort of thing in the world of biotechnology or in the world of science and technology nowadays is artificial intelligence. Now, artificial intelligence integration for faster automated data analysis, improved diagnostic accuracy is of most importance. So that artificial intelligence integration in the software, in the analysis software, in the detection software will aid more and more insights towards the cellular analysis and towards the disease modeling and prediction of our flow cytometric data. Then we talk about the conclusion. The conclusion is that flow cytometry is an essential tool in medical biotechnology, providing rapid, sensitive, and specific cells on data. So, as I was telling you, what the most important need for science and technology nowadays is first is the rapid, means it should be less time consuming. As soon as fast we get the data, as fast we get the diagnosis, it is better to cure or it is better to prevent the disease. So the rapid or the early detection of the disease is the most important part. Then we talk about sensitivity and the specificity, whether the data is very specific on a particular cell and whether it is sensitive. It can detect the minimum of amount of data or it can amplify the minimum of amount of cellular data into positive results for medical decision. Its applications span from cancer diagnostics and immunotherapy to monitoring transplant patients. future advancement will further expand its role in personalized medicine and precision diagnostics. So I thank you all.